I'd like to begin. I'm Richard Axel from Columbia University. I'm going to talk about neurodegeneration. Now, all of our perceptions, all of our thoughts, emotions, memories, all of our actions are encoded in neural representations, in ensembles of nerves, of active nerve cells that are recurrently interconnected. So that's it. All of cognition is simply the consequence of the activity of an ensemble of nerve cells. That's a pretty astonishing fact. But also remarkable is the observation that these very nerve cells engaged in these essential cognitive activities, these nerve cells have to survive for 100 years. It's a long time. So perhaps it's not surprising that after 70 or 80 years, these nerve cells begin to degenerate, and we observe neurodegenerative disorders. Now, it also follows that with a global increase in life expectancy, this has resulted in an epidemic in age-related neurodegenerative disorders. And the burden is sobering. At present, in America alone, there are over 5 million people with Alzheimer's disease. And the cost for caring for these individuals is close to $200 billion. And it's estimated that over the next 25 years, this number will increase 5-fold. And so in 20 years, the cost of caring for only one form of neurodegenerative disorder is going to exceed $1 trillion. Now, as this problem soars, Big Pharma has significantly reduced their efforts in neuroscience, quite significantly. And the reasons that I have heard include the argument, firstly, that the brain is complex and that significant information about the neural circuits that encode memory, that encode motor action, will be essential before meaningful pharmacologic intervention can be anticipated. Second, they argue the disease often takes 6 decades, 70, before clinical symptoms emerge. And as a consequence, animal models are difficult. They argue that clinical endpoints are rather difficult to define. They're vague and elusive. And clinical trials are lengthy. And you add to that a structural impediment, the blood-brain barrier. So I don't agree with Big Pharma. I also don't agree with the neuroscience community. I don't agree with Pharma's reasoning that these are fatal impediments to the to research and pharmacologic interventions. I argue that Alzheimer's disease, Parkinson's disease, ALS, frontotemporal dementia, they're diseases of neural degeneration. They're diseases of neurons, of cells, and we don't have to understand the neural circuits encoding memory to understand and control Alzheimer's disease, FTD. We don't have to understand the circuit basis of motor action to understand and ultimately control Parkinson's disease or amyotrophic lateral sclerosis. These, I argue, are problems in cell and molecular neurobiology, and we now have the genetic and cellular tools to dissect the degenerative process. So, as I said earlier, I believe that Big Pharma and my colleagues in the basic neuroscience community have been at fault. And they've been at fault both with differing financial motives. Pharma argues that intervention awaits significant additional understanding of the complexity of neural circuits, and this, of course, avoids the costs of research on cellular-based processes. Neurobiologists argue the same. They argue that an understanding of Alzheimer's disease will require a deep understanding of the multiple memory circuits in the brain. And they do this so they can access funds from the Brain Initiative, for example, and give input to their endeavors. So I repeat that indeed I think we are dealing with problems in cell and molecular neurobiology. We're dealing with problems of neurons, and we're now in a position to dissect this process. I can provide you numerous genetic entry points, but I think the panel today will afford this, will provide this for us. And so what I've tried to do is pull together a group of courageous investigators, two and a half, three at universities, and two in biotech. So today I introduce to you Steve Paul, the president and CEO of Voyager Therapeutics. Steve was the head of research at Lilly, scientific director of NIMH, held numerous academic positions. He knows his way around neurodegeneration. Lorenz Studer is a professor at Memorial Sloan-Kettering and is taking perhaps the most global approach to this problem by saying, I don't really need to understand the intricacies of the degenerative process in cells in Parkinson's disease. I'll just replace the cells. And we'll hear from Lorenz. Marc Tessier-Lavigne, president and professor at Rockefeller University, and also a founder of Denali, who brings us a deep insight into neural development, perhaps insight into the question of how it is that you set up a nervous system with neurons that can survive for 100 years, and what it is in that process of normal development and survival that goes awry in neurodegenerative disorders. And Ryan Watts from Denali, formerly of Genentech, who in his early work has approached the physical impediment, that is, an understanding of getting beyond the blood-brain barrier, and more recently has brought his insight to questions, I believe, largely devoted to Alzheimer's disease. So I'd like to turn first to Lorenz Studer, who has courageously asked, given the history of embryonal cell differentiated embryonal cell transplantation in the past, to ask whether I can, with sufficient frequency, elicit the differentiation of neurons in culture in a way that would allow one to safely introduce those cells into an individual whose dopaminergic neurons have been depleted by degeneration in Parkinson's disease. Lorenz?
Sure. So first of all, thanks so much for the invitation, and I also apologize for my voice. I was just traveling, and hopefully the voice is going to last. But so as Richard indicated, we are really on the side where we actually try to replace cells lost in the disease, and basically the idea of cell therapy and how do you achieve that. Now, cell therapy in big pharma has been, again, for many years something that has not really been pushed forward at all due to the complexities, maybe due to the difficult business model, also due to the fact, again, that we don't understand exactly how it works, exactly the argument that Richard brought forward. But we have, I think, very good examples in recent years. For example, we talked a lot about the CAR T cells, that if there are examples where there is promise, suddenly there's actually a big craze moving forward, in this case, to find trained killer cells to kill the tumor. And what I propose is that in a couple of years, we're going to basically have a similar situation where we're going to have trained regenerator cells, and because the technology is getting towards such stage where this is possible. But before I get there, just a little bit of history. What's basically took so long to basically come to that stage. So the main problem was we didn't have the right cell. I think the best way to illustrate that is indeed in Parkinson's disease. It's probably about a dozen different cell types that have been tried over the last 4 decades in trying to replace dopamine neurons lost in Parkinson's disease. And again, the idea is very logical, that by the time you become a neurologist, more than half of those neurons are gone. They are never replaced normally throughout life, so you want to try to get them back. So there were, for example, examples where people used adrenal gland cells. There was a big trial from Bayer using basically retinal pigment epithelial cells that was all the way going to a phase 3 study, but they all failed miserably. And in hindsight, when we look at the preclinical data, we actually can easily predict why they failed quite miserably, because they really didn't survive long term, they didn't function. Now the situation changed a little bit when we got to fetal dopamine neurons. That was work really starting primarily in Sweden, in the mouse first in the 70s, pioneered by Anders Björklund, and then implemented towards the clinic in the 80s by Ole Lindvall and others. And what was different there is actually this seemed to be the exact cell that is lost in the disease, and you can harvest them from fetal tissue, and you can inject them, and the cells seem to survive long term. The problem with that approach was that we didn't really have enough access to those cells. It's not really a defined way to do that, and you don't need to have the current discussion about Planned Parenthood to know that it's going to be a controversial thing to have fetal cells as a routine therapy. Furthermore, when this basically was tried on larger scale, basically didn't work very well in placebo-controlled trials. I could go on for a long time why that is, but it was in part actually again because there were the wrong endpoints chosen. There were patients chosen that were too late stage in the disease. And they were chosen in such a manner because meanwhile people could have TBS, which is not a surgical approach, so only the severe patients were offered that approach. And again, a number of other issues. But what was interesting, even though that was largely a failure, these patients obviously continue living because Parkinson's usually don't die immediately. And they're now patients 20 years out. And so actually the scientists went back and looked at those patients, and it turns out at least about a third of those patients that you can still track, they basically are off dopaminergic medication, no medication. They have normal dopamine levels in the striatum, and at least the movement part of the disease is nearly completely restored, which is quite remarkable in a disease that's progressive. Now, it's not a complete cure, because you probably all know Parkinson's is not just affecting dopamine neurons, but the dopaminergic component is pretty much cured. So now where we are now is we would like to emulate those results routinely, and we clearly don't want to use fetal tissue. And so that took then again more than 10 years of meticulous developmental biology to understand how can we take now a cell such as a pluripotent cell, so these are cells that can turn into all the tissues of the body, and they're basically either embryonic stem cells or induced pluripotent stem cells that are established by reprogramming cells that could be done from anyone in your room. Basically, take a small skin biopsy, reprogram the cells, and have matched cells. So those two sources, they have now been successfully by my lab and by others converted very precisely just in those dopamine neurons, and they can be implanted in mouse, rats, and monkeys, and they seem to work, survive long term, and lead to behavioral improvement. We actually have a model where we can develop them nearly like a drug. So you can scale them, make them in the billions, you can freeze them down, qualify them completely, have large batches, and therefore take away a lot of the uncertainty that you have in past cell-based products. And so I think that's going to be clearly one of the pioneering applications that hopefully is going to go forward now to test the potential of those cells. But we hope that's going to be just one of the models that could be used for many other neurodegenerative diseases. Just one last point, I think that's kind of an interesting basic issue with those cells. When you make those cells actually when they're embryonic or reprogrammed, they are actually still behaving like fetal dopamine neurons, which might be good, because the fetus usually don't get Parkinson's disease. So you put those into the brain, they are probably going to be protected for many years. In fact, patients 20 years later, fetal cells survived. But it also brings a challenge. I think that Marc will come back in his presentation. If you want to use those cells to study the disease, not to make them themselves as the therapy, but to use them for drug discovery, you need to actually understand how to age cells. And so that's now, I think, a big trend. We're getting very good at making not only dopamine neurons, but 50 different cell types routinely. But we cannot so well control the age. We have now some examples where we actually can turn a gene that basically moves the cells forward to a later stage. And what those studies about manipulating age also show us is actually also, I think, quite fascinating. It's actually when you reprogram cells, you can do that from a person that's 90 years old, reprogram back to pluripotent state, make a dopamine neuron, that dopamine neuron is fetal. So you forgot that it came from a 90-year-old person. So that means we need to be able, in the future, and again we're working on that very hard, controlling age in the cells to have aged models for disease. But it also offers quite an amazing opportunity if during reprogramming you can reset age. That means that age is, at least in part, an epigenetic property of the cells that can be reversed. So if you could actually make the cells young, you might actually also, in the very far future, to be able to completely prevent the disease in a very unique way. I think I'm going to go stop here.
So you circumvened a number of concerns.
Sure.
If you use ES cells as the source, as I think you are now operating with, you do have a histocompatibility, but you have an immunologic problem.
Sure. Yeah,
and alternatively, you could begin to think about true precision transplantation using induced pluripotent cells. So how do we go?
Sure. I think there are these two models that Richard alluded to, and like both have significant potential. So we initially chose to go with embryonic stem cells largely for safety reasons. Because going through this reprogramming process, a lot of things happen, and the cells have to be highly selected for the process. And actually the first IPS trial that was done in the world, in Japan, which was in macular degeneration, which is an eye disorder, retinal pigment cells were produced, was stopped currently because of concerns about basically tumor suppressors that basically were changed in those cells. So we think with the ES cells we might have slightly better control over with regard to safety, but that might be a technical issue that's solvable. Now in the brain we are somewhat in a fortunate situation that we know from fetal tissue studies that actually some of those investigators did use no immunosuppression in the brain at all. And cells did survive for 15, 20 years. Now on the other hand, we know that there's an immune response in the brain. It's not completely immune privileged. And so the common, kind of the consensus from most groups is that for the initial injection, where you have a lot of trauma, a lot of exposure of the cells to the periphery, that you want to immunosuppress for about 9 months. But that seems to be enough, and that's maybe a reasonable compromise, because for short-term immunosuppression, the side effects are clearly more acceptable than long-term. And so I think this is kind of the pros and cons. Clearly, going patient-specific would be great from an immunological perspective, but the problem there is, again, the cost is going to be enormous, except if there's a lot of innovation, because this process is very, very lengthy, from reprogramming to differentiation to quality testing, probably about 9 months, and a lot of work for every single patient. So CAR T-cell is actually a piece of cake compared to that. So it would be very, very expensive. And so the compromise that might go forward is actually they're going to be made iPS cell banks. They are HLA homozygous because you can, from the blood banks, you can contact patients where you have HLA homozygous phenotypes that are going to be compatible to many people. And then you make iPS cell lines from those, you very highly qualify them, and then you're going to have banks like blood groups from which you're then probably going to make dopamine cells. Still going to be expensive, still going to be complex. Particularly in the US, we are an outbred population, so you might need probably up to 1,000 cell lines. In Japan, the calculations, you need about 200 cell lines to cover 90% of the population. But these are the different options that are currently discussed about this issue.
The remarkable observation on your animal studies, which is going to relate to other disorders as well, is the fact that in order for these cells to correct motor function, you need both the input and output connections to bear some resemblance to that which emerges.
Yes, so exactly.
But it seems to.
I mean, as compared again to these other cell sources in the past, we really are the opinion that you want to have the exact right cell. So not only do you release dopamine randomly, but you actually know exactly how the regulation of the synapse works. And you can actually show that. So if, for example, made animals that we cured for motor symptoms ourselves, but you have a switch in the cells, just in the grafted cells, where with light you can shut them off. So if an animal that's now recovered, you think it's your graft doing that. You do nothing else in the whole brain, all the host cells are the same, but just the graft is shut off. The animal is again as Parkinsonian as it was 6 months before you injected the cells. So we actually even can go further. We know exactly what synapse it acts. We know which synapse gets potential. It's exactly the way normal dopamine system works. So it's not just the dopamine bag that you put in the brain or a pump. It's actually highly regulated, and that's again what makes me excited for the future, that this might indeed be a better approach.
Now, another use of IPS cells, induced pluripotential stem cells, is to take cells from individuals with mutations thought to be responsible for neurodegeneration and use that as a model system to try and obtain insight into the degenerative process. Marc, can you talk a bit about that?
Sure, but maybe, Richard, if it's okay, I'd just like to build a little bit on what you said earlier about the when you question the pessimism of some people looking at this field and argued that there is cause for optimism, and it's probably worth, especially since Alzheimer's, I think, is really the biggest condition we're looking at. It's certainly the most prevalent neurodegenerative disease, pointing to cause for optimism in the clinic right now with the antibodies that deplete amyloid. The data from Lilly, this is an antibody that Steve helped develop, from Genentech, an antibody that Ryan helped develop, and from Biogen, collectively really start to build confidence that, first of all, we can access and deplete amyloid, and secondly, that this appears to be beneficial. We won't know for sure until the phase 3 trials are completed, of course. These are interim results. But in the aggregate, I think that optimism is growing. The second thing that's worth pointing out, and I'm prompted because of an article that came out today, is that when you look at the clinical trial data, it looks like the benefits are still modest. But there's a study published today showing that even a modest benefit can make a real difference. Some of you may have seen, it was picked up in the popular press, an article in Lancet that looked at taking moderate to severe Alzheimer's patients off of generic Aricept, so donepezil. And a year later, the chances of those individuals going to a nursing home had doubled. And that is a drug that has a modest benefit. So even a modest benefit has a big effect in terms of patients' lives. So just a few things to keep in mind as we look at this and ask ourselves if the glass is half empty or half full. I think we have some signs that perhaps the glass is half full. And the reason to mention amyloid also is because, of course, it was first discovered in biochemical studies, histological and biochemical studies of the brain. But it's really human genetics that that really implied that amyloid is a key bad actor in the brain because of the kinds of mutations that are seen in early onset Alzheimer's disease, and they are mutations that cause an increase in the amyloid load. So it's that the human genetics and that correlation that really have made people focus their attention there. That's a build-up to saying that one of the big advances in the last decade has been the explosion of insights from human genetics in Alzheimer's and in many other neurodegenerative diseases. And with that, we now know many genes which, when mutated, either increase the risk of various diseases, whether it's frontotemporal dementia, Alzheimer's, Parkinson's, or actually are causal, where someone who carries the mutation is destined to get the disease. And to your point about induced pluripotent stem cells, they provide a model to study what the effects of these mutations are. So it's a field of great promise. You can, as you say, take skin biopsies from a patient carrying one of these mutations and derive iPS cells from which you can make neurons in a dish. With the technology we talked about previously, the gene editing technologies, whether TALENs or CRISPR, it's possible to correct the gene so you can have matched cell lines, one with the mutation, one without. And you can even get away from having to get biopsies from patients. You can just engineer in the mutations directly and compare things in a very controlled way. You can make neurons, and then you're sort of off to the races, with one exception, which Lorenz mentioned, which is that the neurons are still very young neurons. They're really embryonic neurons. When you make these iPS cells or ES cells, they revert to an embryonic phenotype. And most of us don't have 60 or 70 years to watch the neurons age in a dish to see whether they will develop Alzheimer's. So there's a lot of effort to see whether it's possible to accelerate the aging process. Lorenz himself is a pioneer in this. Some of the tricks that are used, there's a disease called progeria, where many of you have seen this. These children who, many of their organs, including their skin, ages so that by the age of 10 they look like old people. That's caused by a particular mutation, and if you introduce that gene product in cells, you can age them, as Lorenz has said. So there are various tricks, that and others, to try to age the neurons, to be able to get representative models in a dish of aged neurons carrying mutations, asking what the mutations does, how it causes the degenerative process, and ultimately to get a model to be able to try to block the degenerative process. Even if you can't get an accurate model of degeneration. If you introduce a mutation into one of these cell lines, you can ask what biochemical processes are altered, because often in drug discovery you don't necessarily have to understand all of the details of what happens. If you know there's a causal gene, it causes biochemical changes, it may be sufficient then to find a drug that interrupts that or reverses that, even if the model itself is not an accurate model of the phenotype. If it's an accurate model of the biochemical pathway, that may be sufficient to make progress. So I think we're seeing people push along multiple lines of those kinds of avenues, taking advantage of the explosion in human genetics and trying to make representative models, and even if they can't, trying to exploit them for drug discovery purposes. One of the additional reason to believe that this is a time where we're going to see acceleration in our understanding and acceleration in the identification of additional targets and of additional therapeutics, in addition to the few that are already making their way through the clinic.
Yeah, let me make a few comments just to kind of extend what Marc says, and I completely agree with his optimism. We've learned a lot. Curing or preventing disease is hard. We're going to make a lot of mistakes along the road. But I think in the case of Alzheimer's disease, as he eloquently stated, and Richard, you underscored, the genetics is pretty clear. Going, though, from a mutated gene that we know is dominantly inherited and causes the disease to the ultimate disease, the onset of the signs and symptoms of disease, is a more, how should I put it, challenging process. Even in the case of early onset Alzheimer's disease, where you inherit a mutation in either APP or PS1, PS2, you still have about half a century until you actually will come down with the signs and symptoms of the disease. The other thing we've learned, in hindsight, this is sort of why didn't we think of this earlier, is that the pathology that precedes the loss of neurons in these diseases starts occurring, probably in the case of amyloid at least, 20 years prior to the prodrome, to mild cognitive impairment, which occurs 4 to 5 years before the onset of form-fruste dementia. We're looking at treating with anti-amyloid drugs. The first set of trials took place in patients who had mild to moderate, in fact mostly moderate disease. So they had the disease for 6 or 7 years, and by that time it seems very clear to virtually everybody in the field it's just too late. And so what people are doing now is they're actually they're actually treating much earlier. They're treating prodromal patients. The aducanumab trial that Marc referred to from Biogen, 40–50% of those patients were prodromal patients. They had MCI. The other half had mild. And the Lilly solanezumab data, the only signal that was seen, quite frankly, was in the mild patients. So said another way, I think we're seeing a signal. How big that signal is and whether it's reproducible, we won't know probably to the end of the year. But I think it's all, those studies have already stimulated a lot of excitement and opportunity in the field, because if those things are real, now we can go back, improve upon those signals, get bigger effect sizes in these patients. And, again, Richard, as you said, this is arguably the biggest public health problem of our generation, our children's generation. It's a tsunami waiting out there, as Ken Fraser said this morning. Daryl gave a nice shout out for your program, your BACE inhibitor. If we can just slow progression, slow onset by 6 years, that alone will cut the prevalence by 50%. That would be an enormous contribution to society, but obviously to patients. So I share Marc's enthusiasm. I think the glass is clearly half full, not half empty, and I think we're on the road to actually maybe improving upon what we're seeing so far.
Marc, can I quickly ask, the linkage studies that you refer to clearly define these mutations as cell autonomous mutations, it would seem. That is, but the GWAS studies have identified 20 genes. They implicate cholesterol metabolism, immune function and inflammation, and endosome trafficking, I think, are the three main categories. So especially with immune function, how do you try and replicate that phenomenon in a dish?
Well, that is clearly a taller order, which I didn't get into the details, obviously.
Well, I know who I'm speaking to.
The IPS technology is particularly useful for cell autonomously acting genes. You can make a mutation in that cell. You can make neurons from that cell. You can have clusters of them. In principle, you could model other phenomena by you can certainly make microglia in a dish. You can generate other components, whether it's glia, microglia, other kinds of neurons. So it is possible in principle to model that. I think perhaps in those cases the most fruitful approach, at least in the near term, will be to ask whether in a dish or in an animal model or in some other model, how do those mutations affect the function of the immune cells? At this point, we're still a little bit in the dark, quite a bit in the dark, as to what those mutations are doing specifically. I think what everybody, the first place to look is to ask whether they alter amyloid metabolism in some way, for example, reducing the clearance of amyloid or things like that. But that's just because it's the most obvious thing to look at doesn't mean that that's what is going to be the effect of the mutation. So I think we have to understand how those mutations are conferring risk, which at this point we don't, and whether iPS cells or other approaches will be the best way to assess that, I think, is an open question. Yeah,
although I would say that for 4 of the SNPs, CD33, TREM2, ApoE4, and clusterin, there are pretty good data that suggests that microglial function is impaired, and that may relate to amyloid deposition and amyloid clearance. Could also relate to tau clearance. It's not entirely clear. But there's good in vitro data, in vivo data that's coming from several labs. So it's still early days, but it's exactly, I think, what's starting to pan out.
I'm not quite sure, though, that data really is derived from the in vitro setting, right? So most of that data is in vivo, and I think very convincing. If you go back to the GWAS hits, as you alluded to, Richard, there are about 35 proposed genes greater than 50% of those are either exclusively or very highly expressed in microglia, and arguing, I think, in a big way for therapeutic approaches that may go beyond just APP processing. That being said, they still likely point to the fact that it's really microglial's role in clearing A-beta.
Yeah.
So let's go back to you, Ryan, and I'll ask you first to briefly tell us how we're overcoming the physical barrier, the blood-brain barrier, and then once you've overcome it so effectively, how are you going to exploit these observations in a meaningful therapeutic way?
So I'll answer your question in a little bit of a roundabout way. I just want to go to your original comment on pharma exiting neuroscience and, to a lesser extent, neurodegeneration, and really point out that between the year 2002 and 2012, there were 83 failed phase 3 clinical trials in Alzheimer's disease. Half of those were going after disease-modifying approaches, and almost all of them, with the exception of 1 or 2, were going after A-beta. And the question is, for us as we go all in neurodegeneration, why so many failures? Why is it that we're seeing these failures? Is it the wrong target? Is it the wrong patient population? And what's very interesting, I was just discussing this with Steve, there's very little evidence in almost all of those studies of showing target engagement with the drugs. That's changed a little bit with the BACE inhibitors. I think we're in a new era. Being able to see very nice reduction in A-beta with the BACE small molecule inhibitors. But if you look at that period of time, there was very little evidence of target engagement. And likely, it was a consequence of the blood-brain barrier limiting uptake of drugs, both small molecules and large molecules. And Marc alluded to the Biogen data. And I think what's most impressive about the Biogen data really is the dose response in amyloid reduction. Okay, we don't know if that's really going to correlate with cognition, but for the first time, you start to see a dose response and target engagement and amyloid reduction. And so, we've taken it one step further. We, talking a little bit in past tense and maybe a little bit in present tense, we developed technologies to get large molecules across the blood-brain barrier using the natural transport mechanisms. And so this idea, the blood-brain barrier is this endothelial barrier that essentially restricts uptake of almost everything unless selectively transported. And the idea is to engineer molecules to latch on natural transporters and get them across the blood-brain barrier. And so over the last 5 or 6 years, we've found a way to take it one step further. It was first proposed over 20 years ago, but it's really around how your molecule binds to that receptor. It has to bind just the right tightness, not too tight, not too loose, to basically get it into the brain. And what we're seeing now is that we can get great exposure over 14, 20, 21 days of a large molecule in a primate brain, and that data was published last year in Science Translational Medicine. And with that in mind, now you have all these new genetic hits, really in the last 5 or 10 years, that can take you beyond A-beta and tau, and you can look at regulating microglial function. You can propose ways to go after lipid homeostasis or ApoE pathway. And we're in a very, very exciting time. I think it's a little bit more than the glass half full. Maybe it's 3/4 full. We have so many new targets and pathways. When I started this 10 years ago, they didn't exist. It was A-beta and tau. And I think it's not just in Alzheimer's disease, the same in Parkinson's disease and ALS. ALS, it was SOD1 in the 90s. In the last 7 years, there are 5 new genetic hits, one of which C9orf is the most common. And it's really a new era for targets and pathways. Now what you need is really a concerted effort all in terms of developing therapies. And do not lose track of the basic principles of drug development, which is show target engagement, which I think we learned in a big way in the oncology space when we were previously at Genentech.
So what do you do?
what do I do at Denali, or what did I do? Yeah.
Now you can get it in, and you can get the drugs in.
Yeah, I think that one of the pathways I'm most interested in is we're calling it the inflammatory pathway, but they're basically microglial regulators. I think this is a really fascinating area. It's across all these neurodegenerative diseases, one of the common pathologies is inflammation. You see upregulation of cytokines, and in isolation that may just be a consequence, but the human genetics in Alzheimer's disease actually suggests that it's also a cause. So I think that's probably the most exciting pathway. I also feel like we've known about ApoE4 for a long time. However, there are very few therapies that are targeting ApoE, and so that's an area I think that's also ripe. It's not just in Alzheimer's disease, but CTE, concussion-induced degeneration. ApoE4 is a risk factor. I think that's also a very exciting area to spend time and effort on.
Let me put in a plug also for gene therapy too here. We talked about this in the last panel, and also something, Richard, you said. There are a number of monogenic neurodegenerative disorders that we know about. ALS SOD1 is one. Huntington's is another, where we're almost certain that if we could, in those cases, knock down those genes effectively with antisense technology, we would delay the progression of the disease. And you can now deliver pretty effectively using AAV antisense constructs either packaged in microRNA cassettes, small siRNAs, for example. There's also these sort of loss of function mutations, diseases like spinal muscular atrophy, Friedreich's ataxia, where we know the mutant gene, we know what we have to replace. And I'll just put a plug in for a competitor company. Avexis has announced some pretty exciting data on spinal muscular atrophy, where they're delivering SMN1, which is the missing protein there, using a viral vector, AAV9, an AAV vector, and getting some pretty remarkable effects, albeit it's still early days, and these are uncontrolled studies, but kids are not progressing to become ventilated or any of these other critical bad milestones. So I think this is another possibility. You can vectorize antibodies, for example, get them a bit better across the blood-brain barrier, put them directly in the intrathecal space.
Now, how do you do this? I'm impressed. So the human brain is big.
Right.
It's got 86 billion neurons.
Right.
And I put viruses into mouse brains. I don't do anything. But the work of the righteous is done by others. But it's a Talmudic line. I put viruses into mouse brains 10 times a day, and I can infect a millimeter or two. And now I've got a couple of temporal lobes to deal with. And I've got a mutation, which is a dominant negative mutation in the amyloid precursor protein. What are you going to do?
So you start with a less ambitious problem. So what are we doing? Well, we're trying to deliver a therapeutic protein to a very small region of the brain called the putamen to try to get patients to respond to L-DOPA once they stop responding. We fill up a little area of the putamen. We can do this visually. We're doing this in human Parkinson's patients as we speak. We use MRI-guided stereotactic surgery and interoperative visualization. We use the MRI to guide the cannula and to see where we're filling up. We have a PET scanning technique that's been well developed to look at the activity of the enzyme we're delivering. We use the substrate that's labeled with F18 so we can precisely know whether we're delivering the gene. The other things we're going after are all spinal cord delivery. So we go after intrathecal administration and the cerebral spinal fluid that surrounds the spinal cord. And in very large species, monkeys, non-human primates, and pigs, we can get great transduction. This is not just our work. This is work of a number of independent labs of motor neurons and glia around motor neurons up and down the cord, from the cervical part of the cord all the way down to the sacrum. And we believe that if we can get that good transduction and knock down SOD1 or replace SMN1, as in the case of SMA, we should be able to move the needle on the disease. And particularly in the case of a recessive disorder, we know that we only need about 50% of the protein, and the patients don't become sick, right? If they have one healthy allele from one of their parents, case of Friedreich's, case of SMA. So that's the strategy. We won't know really whether it'll work, but the delivery problem, as was nicely articulated in the gene therapy session, is a big problem, a big issue, and I think you have to start in a fairly less ambitious way moving forward.
So I think we've heard from four, I consider, somewhat courageous individuals who really do have an optimistic feel and an insight into this problem. I maintain, some of them may agree with me, that while it may be of some value for us to understand the neural circuit underpinnings of thought, that that's not necessary to understand the cognitive decline in AD. So I'll open this, unless there's something anyone wishes to add, I'll open this discussion to the floor.
Thank you. This question is for Dr. Watts. We have heard that microglia-induced inflammation could be a cause of Alzheimer's. I personally believe that more than other culprits. Can you tell us if there is this inflammation going on? Can we identify cytokines and other signs of inflammation? Is it possible to determine that?
Yeah. So I think you're pointing out one of the challenges with just drug development in general, which is the necessity of biomarkers to show target engagement. And as it relates to neuroinflammation, the short answer is you can. It's difficult. So in CSF, you could, in cerebral spinal fluid, which the brain is bathed in, you can pull out CSF and you can measure these cytokine levels. What I think is really interesting, and it has made some progress, are PET probes to allow you to image inflammation. There are these TPSO PET probes that allow you to see inflammation. The challenge there is that they really work in massive inflammatory situations like MS or stroke. They're not so effective yet in Alzheimer's disease. And so I think part of our challenge in drug development will be getting the biomarkers that allow us to really show this target engagement. But that's exactly what you hope to see, is you develop a drug, you know it's the dose, and I think something we haven't hit on yet, Richard, is you know you're going after the right patients because you're either using genetic biomarkers, imaging biomarkers, or CSF biomarkers. I think we learned that, in a very interesting way in the oncology space. So with Herceptin in breast cancer, when Herceptin was originally developed, it failed in clinical trials in all comers. But actually, the HER2 positive patients had an incredible response, and it just got better and better the better we got at basically identifying that patient population. We treat Alzheimer's disease as this sort of massive, all patients, all comers, and of those 83 failed clinical trials, there was no pre-selection of a certain patient population that may or may not respond. So as we develop these specific immune modulators, we should also look for either the genetic or the biomarker evidence of those pathways being activated. And you can use it to select patients, and you can also use it to show that your drug is engaging the target. And I think we're getting close. Some of the targets are better than others to allow us to do that.
It should be possible, at least in mouse models, to unequivocally demonstrate the site of action of the mutations.
So
right,
absolutely.
Bone marrow transplants, for example.
Yeah, so absolutely, and I think that has been done. I think in our animal models we're getting very good now at seeing a dose relationship. We can measure, cytokines, and in our experience, in every neurodegeneration animal model that we've measured, we've seen the canonical cytokines massively upregulated as the disease progresses.
Hi, I'm Robert DeVita from Mount Sinai, working on small molecule drug discovery, and we're working on projects that would lead to cell proliferation in specific tissues or activation of proteins, and I think these would be applied well also to neuroscience, so going to activation of proteins or in cell transplant you need to have those cells proliferate or activate. So how do you balance the safety issue that our pharma colleagues and our colleagues in oncology would have for activating proteins or putting cells into places and having them proliferate versus causing negative effects in those areas?
Lorenz.
Sure. That's obviously a particularly relevant concern for what we are trying to do with cell transplantation. And so you can solve it in many different ways. One is just by qualifying the product you put in. If it's a fully defined product, you can actually put cells in that are already post-mitotic. So that dramatically reduces the risk, because there's really pretty much no risk from a post-mitotic neuron to form tumors. But what you're also alluding to is obviously many groups think you can actually maybe stimulate endogenous cells or the endogenous proteins under those conditions. And as you said, that's obviously quite challenging, because sometimes you think you know that you stimulate from a precursor population that then turns into neurons. But a good example of that is was tried for in seizure models, and actually makes things worse, because the cells, yes, they proliferate, and then they make neurons, but they don't make the appropriate neurons in these conditions. And so I think it's clearly something which is relatively difficult to approach, because you need to know exactly for each case what you try to achieve and then test for that. And again, in the models like Alzheimer's or Parkinson's, I would be very skeptical that in the short term you can find a way to stimulate those neurons, because again, that would have to be it would have to be reprogramming, because normally that doesn't happen. It happens only, as Richard said, early in development, and then you have the cells for 80 years. So, but there are actually groups that literally going to try to reprogram glial cells into neurons. And again, that's still very, very early, and we're not sure that that's going to work, but it's definitely a potentially interesting approach.
Yeah,
That's all.
the issue with Alzheimer's and cell replacement is a complicated one, because it's thought that the hippocampal neurons have a set of connections whose weights, whose strengths reflect the experience of the individual. And it's very hard to replace experience.
I think Nick had it.
Yes.
So first of all, thank you. It's great to hear the optimism. The question I'd have, though, is a little bit around, you articulated, Ryan, the scary nature of the history of drug development in this area. So a couple of questions related to that. It strikes me, obviously this is extremely complex, but it does strike me that our system's not quite set up for success here. And I'd be curious about your thoughts as it relates to the regulatory environment. Do we need to have change that incents this or allows different pathways specifically for what is clearly the most seismic challenge we have in science, and that relates to all of us? So from a regulatory perspective, or other ways to incent big pharma companies to take risks in this area when they've been hurt dramatically. I see 2 startups here who are funded reasonably well, but nowhere near where you need to take it to. Yeah. So how does this all, the ecosystem strikes me as not being quite ready to receive the challenge. I'd be curious about your thoughts along that dimension.
You've been everywhere.
Yeah. So I think the ecosystem is getting ready. Remember I mentioned earlier that the earlier you're likely to treat, certainly with an amyloid therapy or a tau therapy, the better the results are likely to be, just based on the, on the pathogenesis that we know from very good, biomarker studies that have been done in patient populations. There are now 3 preclinical, large clinical trials. The Diane trial, which is in, genetic, early onset cases of Alzheimer's where patients are genotyped, they know they have an affected parent, it's a dominant disorder. And then Risa Sperling has a very large trial up in Boston called the A4 trial, which is older individuals that have amyloid in their brain and are at high genetic risk. They might be an E4 carrier. They're certainly at high pathogenic risk, if you will, for development. So trying to get patients into trials earlier and earlier. The other little fact that is worth repeating is that in the early bapineuzumab trials and a number of the solanezumab trials, no one ever really tested to see whether the patients actually had Alzheimer's disease. So once they did spot PET imaging, In the BAPI trial, it was really frightening. 36% of the patients, the E4 non-carrier patients, did not have demonstrable amyloid in their brain. So if you're starting a clinical trial and almost 40% of your patients do not have the disease you're going after, you've got a problem. So there's been a lot of learning, and I just point out, I don't want to overstate the aducanumab data from Biogen because there are some issues there. It's only 150 patients, and it was 5 arms. They had 1 placebo arm and 4 doses, 150 patients, and they were able to see almost a statistically significant effect. They did see a statistically significant effect, on amyloid deposition or amyloid burden. So I think these trials are going to get better. I also know there are efforts underway to try to pre-enroll patients to identify cohorts of individuals who meet the enrollment criteria for these trials, and I think that's going to help a great deal as well.
So a year ago on this exact stage, Janet Woodcock was sitting right here, alphabetized, I was right there. And Janet was specifically calling out for using biomarkers to select patients. And I think, that decade of failed trials, it's a great example that Steve brought up that, at least 30% of patients enrolled in those trials will not have amyloid. And so actually we kicked off one of the first Alzheimer's prevention initiatives in Antioquia, Colombia, treating presenilin mutation carriers at age 30 that would develop disease at age 40, and you're using very careful cognitive endpoints. Obviously, they're all they're genotyped, although it's triple blinded. The patients don't know if they have the mutation or not. They don't want to know because they know what the end result is, and it's trials like that in these subsets that I think are we're seeing a new generation of drug discovery compared to the decade ago of just treating all patients. And the FDA was very, very engaged with us in that process and very supportive of running that study. So I think that there is, they have a vested interest in success as well.
Yes. This is a very important discussion you're having. None of you brought up the issue of schizophrenia. That occurs early. It is a devastating disease, and I believe there's a neurodegeneration element to this, but I would like to hear your comments.
Well, the question as to whether schizophrenia is a neurodegenerative disease is a complex problem we could discuss at some length later. There is, in fact, evidence for white matter thinning in schizophrenia, some gray matter thinning, but the real question is whether that thinning, which might be interpreted as degeneration, is causative or a consequence of the disease. And so I personally don't feel there is any compelling evidence to suggest that schizophrenia is a degenerative disorder. But I know nothing.
No, I've spent about 20 years working on antipsychotic drugs and schizophrenia basic research. It's a very difficult, challenging area. It's a very important problem. There's no argument there. I think Richard is absolutely correct. The neuropathology there has been murky at best. And frankly, in my opinion, the genetics have been very, very challenging. Even though we know there's an inheritable component to the disease, there's cross-adoption studies, there are twin studies, monozygotic versus dizygotic twin studies. There's no question there's a genetic component to it. But finding genes that really matter in that disease have, unlike what we're talking about in Alzheimer's disease and the monogenic neurodegenerative disorders, has been really challenging. So we don't have a lot of biological clues as to kind of what to do at this point.
I think we should move on. I'd simply like to thank the panelists for their contribution for tolerating my ignorance. Thank you.
Terrific. Thank you. You are terrific.
I don't know anything.